Okay, so we're moving at a pace with this virus that is really unprecedented in the history of pandemics. And so that video really encourages me because it tells me that there is true value to us working together as a globe, scientists from around the world collaborating, but also even people like you and I who aren't doing any of the scientific research, we still play a part because when we are out and we're wearing our masks and we're trying to socially distance, or when we're staying inside of the house, what we're doing is ultimately prioritizing the needs of society over our own individual needs. Because even though it might be uncomfortable and hot for us to wear that mask, even though it's not ideal for us to be talking to each other via computer screens, what we're doing is prioritizing the health, not just of us, but of also our family members and the people we live with and the people who we would interact with on the way to school and in other circumstances. So it's really a big teamwork effort. And we haven't been perfect. We know that in some states, people have refused to wear masks, and they basically have just said that it's not worth all of the damage that we've done to the economy. So we know that not everybody has been a team player, but for the most part, we've done a good job, and I'm proud of you guys for taking on this challenge of learning virtually because it hasn't been easy. I know. All right. So like I said, we don't have too many notes to take today. But hopefully you have your notebook out, and we're talking about unicellular adaptations. So as I said briefly earlier, unicellular organisms like plants and animals have organs and organ systems that help them perform essential life processes. So you and I have lungs, and our lungs have specific cells that allow us to breathe and allow us to undergo the process of respiration. Plants also have specific cells called stomata that allow them to take in carbon dioxide and then to expel oxygen. Many animals have either an internal skeleton that give them structure and allow them to move, or they have an exoskeleton that also provides some protection to the outside world. So we have different organs and organ systems and different types of cells that allow us to do these things, but unicellular organisms like yeast or bacteria or amoeba, they do not have this ability to specialize. So they require on adaptations to survive. They require adaptations. Oh, there's a large roach. Okay. One of these adaptations is called an eye spot. This one is pretty easy to remember because it's basically exactly what it sounds like. Obviously, these unicellular organisms do not have actual eyes, but they have what's basically a collection of specific proteins that allow them to detect light. And of course, for many of them, they need the light. The light serves as a source of energy for them. And so with this eye spot, they can detect the light and then they can undergo what's called phototaxis. Break that word down. Photo means light. Taxis means movement. So phototaxis means movement towards or away from light. It means movement in response to light. This roach is really, really trying me right now. Okay, sorry about that. So, quick video of an eye spot in action.
The video shows a typical euglenophyte euglena. Euglena does not have a rigid cell wall, usually referred to as a pellicle, so the shape changes as the organism moves.
Okay, I didn't like that video. Let me see if I can find another one. Okay. Well, that's not the best. There are specific videos that show how these cells move towards light. If you flash light on them, then they'll move towards them. But this one didn't really show that. That's okay though. Okay, another cellular adaptation is called a pseudopod. The P is silent. The first P is silent. Pseudopod. And a pseudopod is a temporary foot-like projection of the cytoplasm and cell membrane that allows an organism to grab and ingest food. So essentially, the cell will extend itself, it will extend its outer wall, cell membrane, to it will extend its cell membrane to capture food, bring it in, and then digest it for use for energy. We can break that word down as well. Pseudo is a prefix that means fake. Like a pseudoscientist is a scientist who focuses on science that is not real. So we can think about the science of eugenics, meaning that certain people are just born naturally smart. Of course, that is not really a real science because it does not, well, that's a different conversation. But pseudo means fake. Pod means foot, like a podiatrist, someone who focuses solely, a doctor who focuses solely on the foot, the bones of the foot, the muscles, the tendons of the foot. So a pseudopod is a fake foot. So we can see these projections coming out. Okay. Then we also did briefly mention cilia, which are thousands of tiny hair-like structures surrounding a cell, allowing movement and the ability to sense the environment. So again, the ability to sense the environment is important if we think about sternger, which is that overarching theme that we introduced in Unit 0 in the first week of class. Sternger, one of the R's stands for regulation, which means that we've got to be able to, all living organisms need to be able to sense their external environment and then make changes internally accordingly. So these cilia sense the external environment. They can say it's too hot over here, it's too cold over here, it's too acidic over here, it's too salty over here. And then they can also contribute in some ways to movement away from or towards the better environment.
Many cells use flagella for moving around. These Chlamydomonas swim very fast using their two flagella. At higher magnification, the two flagella become visible when a cell is embedded in mucilage, slowing their activity right down. Like many algae, Chlamydomonas has an eye spot which responds to light and affects the way the flagella beat, steering the cell toward or away from it. Many protists have flagella of different length and function. This euglenoid, called Phacus, is swimming in the direction of its longer flagellum. These relatives of Phacus also have two flagella which function differently. One beats in a coil around the cell. The other, as in Phacus, extends forwards. Other algae have more than two flagella. This cell has 8, inserted into a deep pit. It normally swims very rapidly and had to be slowed down to have its picture taken. Large protists like Paramecium swim rapidly using hundreds or even thousands of flagella, or cilia as they are usually called. They are very agile as they search amongst the pond debris for food. Beating of the cilia is coordinated, and the direction of beating is reversed when the cell changes the direction it wants to go. This cell, like the last ones, had to be slowed down in a sticky solution so that it could be photographed. Many protists also use their cilia to create currents for feeding, carrying nearby organisms close enough to be eaten. These algae are too big for this protist, but show the currents clearly. This large protistan cell called stentor feeds like a vacuum cleaner. A circular array of cilia around the flattened end of the cell sucks nearby organisms into a conical mouth where it can check out what it has captured. It is feeding here on green algae. Notice that it is quite selective about what it eats, swallowing some of the captured cells and rejecting others. The cells of some tissues in animals have multiple cilia, like these cells cultured from lung tissue. The activity of the cilia is coordinated so that liquid or mucilage can be moved long distances over the surface of the tissue. Flagella and cilia contain a core of microtubules. Using polarized light for imaging the cells, the microtubules show up as a dark or light fiber inside each cilium.
Okay. So cilia serve multiple functions depending on what the overall needs of the unicellular organism are. Here's a pretty good image.
Are those in water?
Possibly. I don't know exactly what the medium is. Like he was saying in that video, sometimes they put them into these specific media, which are either thicker or stickier than water, so that the organisms can't move as quickly. Okay. And then the flagella, that's the plural version of the word. Singular would be flagellum. These are the tails or the whip-like structures that help a cell move through liquid. And we're going to watch a video specifically of how sperm cells do this. But chemotaxis, if we think about what phototaxis is movement in response to light. So chemotaxis is movement in response to a chemical or multiple chemicals. So this is actually how the sperm can find an egg cell. The egg cell has a specific chemical scent, and then the sperm cells can swim towards that scent. Oftentimes, organisms will have multiple flagella, but sperm cells have one. Okay. Okay, so there are better videos. In fact, I probably will replace that video. But essentially, once the sperm cell finds the egg, it will embed itself into the egg. Keep in mind that a sperm cell has 23 chromosomes from the father. The egg cell has 23 chromosomes from the mother. So once the sperm cell embeds itself into the egg, they create what's called a zygote. And we'll talk more about that later in the class. But once it embeds itself, it loses its flagella. So that tail is no longer useful. So it actually just falls off. And now you've created a zygote, and that zygote has 46 chromosomes. And it will begin to develop into an embryo, and then a fetus, and then a human being. All right. Contractile vacuoles are important because, as we just talked about and Ayana asked the question, most of these unicellular organisms exist in water. And so they need to be able to store water, but they also need to be able to pump it out when there's too much. So you can see it briefly right here. There's some water movement, some storage going on, and then it gets expelled. This is primarily to maintain pressure. And also water is important and is essential for respiration, which is the creation of energy, cellular energy. So that's important to note as well. Not sure why this is. So you can see, yeah. So this is 250 times magnified. Now 1250 times magnified. So it almost looks like the fluctuation of the lungs, constantly taking things in and then pushing it out. We can also see a pseudopod forming right there as well. All right, so that's it for the synchronous portion of class. For you guys, we did have those issues with the previous assignment on Friday, which was called labeling cells. I did fix it and I uploaded a new assignment, and so it's on day 3 now. So it's still called labeling cells, but it should say labeling cells day 3. And then there are other assignments as well. So we still have about 30 minutes in class. I will mute myself.
Mr. Rudd, I went to my Google Drive to find my biology folder, but there's nothing there. I know I did it because I turned it in and shared it with you. I just don't remember what platform it was on.
What I would do you remember what it was? Well, let me see if I can find out. If you just search, you named it Ayana, Aaron, biology folder. Not sure why you put the comma there, but if you just search that,
I think I'm doing something. No, nothing. My biology folder hasn't showed up. Did it get deleted?
no, because I can still see it.
I can't see it on my.
Yeah, you're still listed as the owner of the folder, so I can't share it with you. Give me 1 second. Okay. So Ayana, you see if you can open that link that I just sent to the chat. You should be the only one. You and I should be the only one who can access it.
I can see it now through the link. I don't know what was wrong before though.
So, I mean, it should still be in your drive. Just make sure that you've saved it. Just double check and make sure that you've saved it one more time.
How do I do that again?
If you Okay, give me 1 second. So if you click on the arrow, I'll share my screen. So you're looking at your folder, Ayana Aaron biology folder. This arrow right here, if you click on it, you should have the option to either move it somewhere or add a shortcut. It should say something like that.
I keep clicking on the drop down thingy, but nothing's dropping down. Isaiah, hush.
I'm not sure what to do. So have you tried to refresh the page? I feel like that's the best advice that I can typically give.
Well, thank you for trying to help.
Have you refreshed the page?
I'm doing that now.
Okay.
No, I think it might be the internet though. So
Okay.
I'm going to actually set up this hot spot thing right quick.
Okay.
Mr. Red,
Yes, ma'am.
can I have permission to leave?
Oh, yes, it's 11:35. Thank you. So you all are good to go.
Sir,
Yes, ma'am.
I don't, the same test, I don't think it's working again. I'm trying to press the image, it's still not doing it.
The labeling?
Yeah.
Okay, give me 1 second. Everybody else, you all are good to go. Is it the assignment that says, hold on. Labeling cells day 3?
Yeah.
Okay, give me 1 second. See if I can. So, Alicia, can you see this? Can you see my screen?
Yeah.
Can you use this to answer it?
Mm-hmm.
Okay. Stop recording. Let me know when you need me to go to the second question. No rush, though.